So we're going to be finishing up probably our chapter 3, part 2, all of the anatomy, the organelles of the cell. I think we're going to, if we finish a chapter, you know what that means. Chapter test.
Yeehaw.
Yeehaw. I think we're going to go ahead. We won't be doing it tomorrow. I don't like to do a test right the next day. I like to work in a bit of review. So I think we're going to do it Friday. We'll do it like we did last week. I'm going to open it on Friday during class time. It'll open for you guys at 8:40. It will close at 9:55. I honestly don't know how many questions are on it yet. That's on my to-do list today. So we'll have to write some good test questions during class today.
Will it be like as last time where we didn't have to come on to Zoom?
Yeah, so you will not have to come on to Zoom. I'm going to give you the whole class period to log in and do the test. So we will not be Zooming on Friday, and that'll give you the whole class period. Same old though. If you log in 5 minutes before the end of class, the test will close at the end of class. So make sure you log in with plenty of time. Your test yourself questions will be due. I'm going to open those up today. So you'll be able to submit those into the Moodle anytime between now and the end of class on Friday.
And to test yourself for what chapter again?
For chapter 3. Chapter 3. And then when we finish this, we're going to get started on chapter 4, which is all of the physiology. How does stuff move in and out of cells? None of the stuff we cover on chapter 4 will be on the test though. The test will only be on chapter 3. And I do have a chapter 3 testable material PowerPoint on my website, not on the Moodle, but on my website. So if you go to the same place, I've condensed down, because I know with these 2 PowerPoints, even though these 2 PowerPoints are relatively short, put them together, it's like 70-something slides. So I have narrowed it down for you. So there is a chapter 3 study guide on my website. As well as a bunch of really helpful videos that might help you prepare for the test. Any comments, questions, concerns about that? Fabulous. All right, so just to do a wee bit of review, we were talking yesterday about our cytoskeleton. Oh, speaking of skeleton, we got a cow bone right there. What is that? That's a humerus. Humerus in his front leg or his back leg? Front leg, right here. Anyway, so in our cells, they do have skeletons as well. We have our microtubules, and remember tubules are made of tubulin, and they are the largest structure in the cytoskeleton. They're the papa bear. We have our mama bear, the intermediate fibers, and those act like the guy wires. They add kind of opposing pressure to keep the structure of the cell. And then we have our microfilaments, which are located very close to the cell membrane. And these are made of a protein called actin that can be disassembled and reassembled really, really quickly. And it allows the cell to change its shape pretty dramatically. So there's our microtubule. The thickest and largest of the fibers, they're used to anchor and move organelles around the cell. Our intermediate fibers act like guy wires. And the different names here, now these 2 names, tonofilament and neurofilament, all that's saying is what kind of cell are they in. Tonofilament is an epithelial cell. It's still an intermediate fiber, but it's in an epithelial cell. Neurofilaments are going to be found in nerve cells, nerve. And then finally, our microfilaments, the smallest of our cytoskeleton, located near the cell surface, and these help to change the cell's shape. And here we have our picture. Get my laser pointer. So here we have our microtubule. Here's another one. Here we have a mitochondria anchored to it. Here we have some endoplasmic reticulum anchored to it. Here I have my intermediate fibers helping to stabilize the cell, and then here woven near the cell membrane is my microfilaments, and those help to change the cell shape. Then we started talking about our organelles. Organelle, by definition, means little organ. The first one we started discussing yesterday was our mitochondria, the powerhouse of the cell, right? I feel like my whole body needs mitochondria right now. I need something.
Are you feeling better than you did yesterday?
I am feeling better than I did yesterday. I got a lot of my homework done last night. I have one quiz and one discussion, and then I can start doing my final projects. But I'm in a good place as far as my school is concerned. But yeah, I am feeling much better. Thank you for asking. So we know that the mitochondria started out as a prokaryote. It has its own DNA, but does not contain a nucleus. It can divide by fission and multiply itself to provide the cell with more energy if needed. And the location of that energy production, which is in the form of ATP, anyone remember what ATP stands for? Adenosine triphosphate. And it's created here in these little caves, these little crevices, which are called the cristae. Cristae is also where the oxygen and carbon dioxide exchange. So they also kind of function like our lungs.
And so did I say that it produces up to 95% of the energy that it feels?
Yes. Let me move my zoomers here for you. There you go.
You said the what is created in the cristae?
The energy, the ATP.
And it's the dark parts on the
It's
side, right?
these
Yeah.
these little waves, these little, I don't know what to call them.
The grooves.
The little grooves, yeah, the cristae. So that's where the energy is produced, as well as where cellular respiration takes place. So obviously, very active cells are going to have more mitochondria, right? The more active you are, the more energy you need. Great example is our heart cells. Heart cells are probably some of the most active cells in our whole body, hopefully, because if they stop being active, well, we're in a little bit of trouble. And they're so active because every single muscle cell in the heart has to communicate, and they have to contract at the exact same time, at the exact same rate, every single time. If I need more energy, as I said, the mitochondria can kind of pinch themselves off. It's called fission, and create a new mitochondria, kind of like cutting a worm in half. And you get two worms. Technically no, but you get the idea, because the worm will actually kind of not become two worms.
There should be one to split.
This is one in split, but this will become two fully functional mitochondria. And they'll be located where the cell needs the energy. That's kind of a no-duh. I think we covered this yesterday, didn't we? Yeah. Now, the one thing I'm not going to do on your test, I am not going to do this to you. I'm not going to have a photo and make you label it. Huzzah. But on here, you can see there's a mitochondria and there's a mitochondria. So there's our cristae. And the shape of our mitochondria are going to be elliptical, kind of smooth. And then right here, cristae, and that's how you spell it. And that's the site of our ATP production and our cellular respiration. All right, any questions about our mitochondria? So the next one we went over is the ribosome. This is the most common organelle of the cell. That sounds like a great test question. Which of the following organelles is the most common found in the cell? Ribosomes. Doug, make a note. What the ribosomes do is these are the little factories of the body. When we have a need to make a protein or make something that the body needs, We copy it down from the nucleus. The DNA goes, okay, here's the recipe. Sends the little RNA out of the nucleus. The RNA finds a ribosome. So if this is my RNA, my ribosome comes down, blah, 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 and then feeds the RNA through it and reads the recipe and starts making the protein. Ribosomes can be attached to our endoplasmic reticulum, where they will just stay, or they can just be floating around, just floating around the cell. And so these are the things that will read the RNA and make different proteins. They'll just kind of grab amino acids out of the sky and assemble them as they've been instructed by the recipe. They're pretty neat. Our endoplasmic reticulum is an extension of the nucleus. And there are two forms to this endoplasmic reticulum, or ER. We have the rough ER, which looks like it's got these little knobs on it because those are the ribosomes. Those are where our ribosomes are created. And we have our smooth endoplasmic reticulum, which is ribosome-free. The smooth endoplasmic reticulum acts very similar to our kidneys. They help to detoxify things in the cell. If you happen to be someone that has a few burpers, the more burpers you have, the more smooth endoplasmic reticulum you will have. Adult beverages will change you on a cellular level. I find that fascinating, that you can change yourself on a cellular level by a nutritional decision. I use the word nutritional loosely, obviously. It's something you are ingesting.
You said the smooth endoplasmic reticulum acts like a kidney?
It acts kind of like the kidney because it's a detoxifier.
Not right, that was the word.
Alcohol is a toxin. Now, obviously, our cats and dogs hopefully not drinking alcohol. Hopefully. But this is a kind of a good analogy. So alcohol is a toxin. I think we can all agree to that. The more alcohol you drink, your kidneys are actually filtering it out. Your kidneys are like, oh my gosh, look at this. This toxin is getting higher and higher concentrations. We need to get it out. And then what happens after you've had a few burpers? You got to go to the bathroom every 5 minutes. You got to pee, break, what is it we call it? Break the seal.
Yeah.
And then you got to pee every 5 minutes. That's because you have altered your kidney function. Your kidneys normally retain all that fluid. And now they're like, nope,
Get it out.
get it out. The smooth endoplasmic reticulum's job is very similar in that it is supposed to detoxify. So the more alcohol that you drink over an extended period of time, the more endoplasmic reticulum will be formed. This guy right here is why we have tolerance. So when you first turn 21, you have that first, I'm kidding myself, but your first beverage. Don't promote underage anything. Your first beverage, and you're like Amy Farrah Fowler from Big Bang Theory. You take like one sip, you're like, I'm drunk. Well, cut to, many years later when you're a seasoned professional. Now you can ingest a lot more with much less effect. It's because of this guy, because of the smooth endoplasmic reticulum. Where does this apply in veterinary medicine? Medications. Medications can also have this effect, which is why some medications over time will lose some of their effect. Things like phenobarbital. Phenobarbital is used in our animals to control epileptic seizures. But over time, guess what happens? We increase the endoplasmic reticulum. The phenobarbital, which once worked great, now that they have a tolerance, and that same amount is not working as well, and we may have to increase the dosage in order to obtain the same results. So although I'm using alcohol because that's something we can all kind of relate to, in our animals, medications can alter them on this cellular level, which causes them to build up a tolerance against medications. Chronic pain medication, like Rimadyl. Some of you may have your animals on Rimadyl. Carprofen is the generic name. The same thing as like our regular NSAID, non-steroidal anti-inflammatory drug. Ibuprofen. Before 2 would do the job, now you have to take 4 to do the same thing. It's all because of the cellular change in the smooth endoplasmic reticulum. So our rough ER is where we have production of protein by this little army of ribosomes. That's what gives it that bumpy braille-like look. Smooth ER stores different fats and helps to create new fats. Synthesis is the word that we use to create. The smooth ER in different organs, for example, the smooth ER of the cells of our liver, because remember, every single cell of our organs, be it your kidneys, your liver, your spleen, your heart, all have the same makeup. They all have these same organelles. And so in your liver, this is what we were just talking about, the cells of your liver that break down drugs will develop the tolerance to the drugs. And so these cells, detoxifying cells, really do assist our big system organs to do the job that they're supposed to do. And so that's our endoplasmic reticulum. I'm sorry, I'm in your way. Tell me to move. Oh no. Horse down. Buddy. There we go. Move you over here. I'm just destroying the place. So there's just another picture. Here's our rough ER. There's all our little ribosomes. And then our smooth ER, detoxifying. All right, so now we have found the rough ER. We've located the appropriate ribosome. We have fed the RNA through the factory. We've created a protein. Now what? We're going to ship it. The Golgi apparatus is the FedEx UPS store. The protein is sent to the Golgi apparatus. The Golgi apparatus packages it, puts a little shipping label on it, and sends it where it needs to go. It's either going to send it to another place inside the cell or ship it out of the cell altogether. So the Golgi apparatus is our distribution center. It also helps to create different multiple sugars, polysaccharides. So we get these little sacks, these little envelopes. In fact, that's kind of what they're referred to as envelopes. And they take it to the Golgi. Golgi takes a look at it and says, okay, where is this supposed to go? Packages it up appropriately, and then sends it where it needs to go. So it really acts like a FedEx store. It'll modify it, it'll package it, sometimes it'll add some sugars, add some carbohydrates, those kind of mimic the shipping label, and then poof, off it goes. That's it. That's the Golgi. I'll give you guys a minute. Furiously writing. How far did you get into your bones lecture yesterday?
We went to the
You went to what?
skull.
Oh, you went through the skull?
Yeah, and then they
Okay, did you go through like your compact bone, your cancellous bone, your epiphysis, your diaphysis?
Yeah.
What was that look?
I was like, I don't remember those. Then when she said that one.
Yeah, epiphysis are the ends of the long bones. Diaphysis is the shaft. Epiphysis, diaphysis, compact bone.
As we're starting to get further in, I kind of like confused some of your work and some of terminology that I have to like really pay attention to dividing my work.
Okay.
Because they go hand in hand.
Right?
What is the ones that come up here? I forgot. It's like.
Zygomatic.
No, that's
Oh, the maxillary.
Yes,
Oh.
yes.
The maxilla is your upper jaw,
Yeah,
mandible,
that's my.
and then your cheekbone is the zygomatic. There's a picture of our Golgi apparatus, and here, I love this picture, because here we've had, here's our little ribosomes, making stuff. We're gonna package it up. Okay, we're sending it to the Golgi. So there's our little traveling, it's actually called a transport vesicle. Vesicle is just a container. Send it to the Golgi, and the Golgi checks it in, decides, what do we need to do with this? What do we need to do? And then if it needs to be shipped out, it'll package it up appropriately. Boo, boo, boo. This vesicle binds with our cell membrane and releases the proteins out of the cell into the body for use. So you can see my analogy of it being like a FedEx is really pretty accurate. Lysosomes. These function very similar to your stomach in that they have an enzyme that will break stuff down. Lysosome locates foreign invaders. So we have these, I can't believe that this, all of this is happening in one cell. And every single cell is, all of this is happening. So we've got these little patrolmen, they're little empty jail cells called vesicles. And if I find a foreign invader, or if I find something that needs to go, I just go blur and I hang on to it and I put him in jail. The lysosome will then come on over and fuse with the jail cell and then release those enzymes and disintegrate, digest essentially whatever it is was trapped. And so they function very similar to our stomach. It is considered the stomach of the cell. These guys can also, though, trigger. Oh, and when these guys trigger, we got a big problem. Because what they'll end up doing is they will kill the cell. So when the cell is ready and is no longer able to function, because cells are not, they're not infinite. But what happens is the lysosome will just release all of his enzymes into the cell and essentially break down the cell from the inside. It's called autolysis. Auto meaning self, lysis, breakdown. And so he'll just go, and then the cell will break down and a new cell will come and replace it. So
Increase in numbers of cell ages, what does that mean?
So as a cell gets older and older and is now getting ready to be taken out of circulation, it increases the number of these lysosomes, because then when it's signaled, the lysosomes all release at the same time. But we need to make sure that we have enough lysosomes to disintegrate and break down the cell. And so as the cell is aging, it'll increase the number of these lysosomes. And then when the signal is given, all of the lysosomes will open at once, and that causes the self-breakdown of the cell. So you can really see, now obviously, is this something you can see on a regular microscope? Of course not. No. Electron microscope maybe, or one of the more powerful microscopes than what we have here. but you can determine the age of a cell by the number of lysosomes. The younger the cell, the less we have, because their function is only there to break down foreign invaders and debris. As the cell ages, we increase the number of these lysosomes in preparation for the breakdown of the cell. So that would be a great test question. Which of the organelles increases in numbers as the cell ages? Lysosome.
So it's sort of like a star maybe, and they get brighter right when they die.
Yeah, if that's an analogy that works for you, absolutely. You're going to get more and more and more and more in order to get the breakdown of that cell. Because just the few that we're using to break down debris, they may be able to release their enzyme, but it won't be enough enzyme to fully break down the cell. We need full breakdown of that cell. Because what we end up doing, what do we do with a cell that's been broken down? We recycle what we can. So we'll go through and we will pick out what we can, and then the rest is just fully broken down. So that's our lysosome. Any other comments, questions, concerns about any of that? Yeah.
I was thinking, what did you say the smooth orders? I think it's necessary.
So the smooth ER helps to detoxify. It helps to detoxify the cell. How are you guys in Zoom land? Are you guys with me? Everyone's okay? Everyone's on the bus?
Oh, I forgot that you have us hidden. We're good. Or I'm good.
Fabulous. Yeah, I've got you guys minimized, so I can't see you. So if you have questions, just go ahead and unmute yourself. So here's how the lysosome, just kind of a picture for you little visual learners. Here I have my vesicle, right? And I've trapped something. Here, my lysosome, here's my lysosome, and here's all the little digestive enzymes. It comes over and it fuses with the vesicle and breaks down the debris. And then when this has increased in numbers and the cell is ready to be itself broken down, instead of fusing with another part, I'm just gonna release it and that'll break down the cell itself.
How long does a cell live?
Depends on the cell. Like for example, your red blood cells live on average about 120 days. But yeah, it kind of depends on the cell. It also depends on what you are doing to the cell, right? Like if you are not providing it with appropriate nutrition, if you're taking in excessive toxins, some burpers, if you're, taking a lot of Tylenol, which will kill your liver, other medications, which can affect the kidney function. So it really depends, but on average, I would say between 90 and 120 days. And that's why you can only donate blood, if we're gonna use our red blood cells as an example, because the cells, the whole blood that you are giving, it takes about 120 days to replace and replenish that blood. So, great question. All right, so the next one is our peroxisome. These ones are cool. These peroxisomes, we find these primarily in liver and kidney cells. And basically, they are also helping to detoxify. The way that they detoxify is we have these things called free radicals. In a nutshell, now obviously I'm simplifying this to the nth degree. Whenever you have any kind of cellular damage, when do we have cellular damage happening to us? All of the time. Like right now, all of the time, the fluorescent lights are causing cellular damage. The pressure on the capillary vessels in your buttocks, cellular damage. You go out into the sun, even just to walk to your car, cellular damage. Whenever we have any kind of cellular damage, we've got these little free radicals. Free radicals want to go around and create more damage. So what we need to do is we either need to remove them or stabilize them. Those are the only two things we can do. Stop them from continuing the damage. It's almost like a domino effect. Peroxisomes are going to remove the free radicals. And basically the way that they do this is they convert these free radicals into something called hydrogen peroxide. Have you guys heard that term before? You know the stuff. It's under your sink. You get a cut, you pour it on, it goes all foamy.
If there's bacteria.
If there, yes, and interacts with oxygen. This peroxisome, and that's why I think of it peroxisome, peroxide, converts the free radical into peroxide, but we definitely don't want hydrogen peroxide swimming around our cells. And so the second step is that, so the first step is we convert it to a peroxide, and then the second step, we convert that peroxide into water. I know. And this peroxisome is what does that. So anytime we have any detoxification, these peroxisomes are what are preventing further damage from happening by removing the free radicals. Step one, they turn the free radicals into hydrogen peroxide. And then step two, they turn that hydrogen peroxide into something a lot safer and something we need desperately, water. We can also stabilize our free radicals. We don't have to just remove them. Does anyone know what happens to stabilize free radicals? It would be something that you might be able to get from a lovely glass of red wine. Antioxidants. Antioxidants stabilize and bind with free radicals to prevent them from creating damage. And then our peroxisomes remove the free radicals altogether by converting them twice, once into peroxide and once into water. So, and again, all of these things that I've talked about for the last 2 days, all of these processes are happening in every single solitary nucleated cell in your body.
Crazy.
It's an entire universe in a single cell. Now, are all of your cells nucleated? No. Give me an example of a non-nucleated cell. One we were just talking about that lived 120 days.
The red blood cell.
Red blood cells do not have a nucleus. This is happening in nucleated cells. Non-nucleated cells don't have a nucleus, and they don't have these organelles. Let's think about this for a minute. Why? Why would it not be good for your red blood cells to contain all of this crap inside of it?
Overproduction.
So red
They gotta
blood
fight off infection.
So what red blood cells do, red blood cells are an 18-wheeler tanker truck. That's it. They transport stuff. They go to your lungs, they drop off a shipment of carbon dioxide, they pick up a shipment of oxygen, they'll pick up some glucose, they go turn and burn to your cell, drop off the oxygen, pick up a shipment of carbon dioxide, turn and burn, go back to your lungs, over and over and over and over. That's all they do. So if you imagine the little red blood cell as an 18-wheeler tanker truck, And all of this is taking up 3/4 of the tanker truck space.
You can't go there.
Not only can they not go fast, but how do I pack in the enough oxygen and carbon dioxide? It's all a space-related thing. I don't have a nucleus and I don't have any of these organelles because the red blood cell is simply kind of passively moving with the blood flow, picking up the oxygen, dropping off carbon dioxide, or vice versa, depending on where it is.
So then you would consider that the package being moved along.
Yeah, all he does is deliver stuff.
Gotcha.
All he does is deliver stuff. And so if the cell is the empty 18-wheeler, the shipping part, I don't want a lot of stuff in there already because then it just takes up space. And then I'd have to have these ginormous red blood cells in order to transport the amount of oxygen that I need. So I don't need all of this stuff. I don't need it. So I don't have it. And so red blood cells, if you think of it now, are they truly empty? No, of course not. But for our mental analogy, they're empty because we got shipments that we need to fill up. So not all your cells have all of this going on. The majority do though. If it's a nucleated cell, all of this stuff is happening inside of it. And I find that just, I can't really wrap my brain around that. Next we have inclusions. These are just little voids. These are just little areas. And depending on the size of the void will determine what it's called. So you have vesicles, vacuoles. These are all just little voids. Just tells you what the size is. A vesicle is very small. A vacuole is very big. We have some white blood cells that you can actually see the vacuoles on a regular microscope. We have this one white blood cell. He's my favorite. He's a little bit bipolar, a little schizophrenic. He is. His name is monocyte. And what happens is when we have stranger danger, the body, so he's truly like Brent Banner and the Hulk. Mm-hmm. So when this cell is in circulation, he's just like popping them on into it. I don't know why he's English. I don't know why he's got a top hat, but hey, it happened. Suddenly we get the whoop, whoop, stranger danger, and the immunity group goes, hey, Hulk smash. And the little Brent Banner cell goes, that's my secret, I'm always angry. And he wriggles out into the tissue, Hulk smash. And what he's got is these ginormous vacuoles, these ginormous, they almost act like tummies. And he just goes around and goes, and he just absorbs all of the foreign invaders. And then he will either disintegrate them in the vacuole, or he'll sacrifice his life for you. And the foreign invader will kill him, killing itself at the same time. So this is our vacuole is just a void, and it has a variety of different things that it can do.
And that's what happens over there.
That is what happens. Monocyte, you got five battalions of your body's military. Monocyte is one of them. And so monocyte goes in and engulfs. But the problem with monocyte is when he turns into Hulk smash, he smashes everything. He is killing foreign invaders. He is killing friendly fire. He is gobbling up dead cells. He's just doesn't see.
Gets a little out of hand.
He gets a little out of hand. And so where we have really, really, really big problems is when he gets an incorrect signal, Hulk smash, and he's still in his circulation with all of your healthy cells. And he just starts busting stuff up. So, but my whole point is we can look at monocyte under the microscope and physically see these little voids, these little vacuoles, because they're so large in that particular cell. Centrioles. We're going to be getting into centrioles quite a bit in our next chapter when we start talking about the dreaded M word, mitosis. It's okay. I got you. I got you on that. And so what centrioles are, is they are found in pairs. And basically what happens is when a cell is getting ready to replicate itself, because that's how your cells create a new cell, is they go through this thing called mitosis. They make an exact replication or an exact copy. So I've the magic is happening, the magic is happening. I get these two centrioles that line up, and then they go to opposite ends of the cell, and they spew out these little fibers. These are called spindle fibers. And then they basically just do a tug of war, and they pull the cell apart until you bloop, have a brand new cell. So that's kind of their role, is mainly during cell division. They have these little spindle fibers, and then they just go, and they pull it apart, and then you get your daughter cell and your parent cell, and you have an exact copy. So now the parent cell can release its lysosome, disintegrate, but we have a cell to replace it. These centrioles can also be where our cilia and our flagella are attached to. Remember, cilia was the sea anemone, flagella was the forward propulsion. Those are centrioles. I got a great video I'm going to show you guys with the next chapter, showing you the cell division. Everybody okay?
Well, the last one.
No worries. Plenty of time. I don't want to rush you. I have to do a stupid Zoom discussion. I'm dreading it. I hate it so much. So this is for my dermatology class. Usually we have written discussions where they ask a question and you post an answer and then people comment on your answer. This one, we have to discuss any of the diseases that we've talked about the whole term, but we have to record it. I know. And then post our recording so that other people can watch it and comment. I'm like, just let me type it out.
So you do your own prior auditions, you practice by yourself? Yeah.
Well, but I'm one of those that, you know, that I can talk like crazy in front of you guys with no issue whatsoever.
But when you're kind of.
But when I'm looking at myself talking, oh my gosh, those stupid PowerPoints that have to have audio embedded into them, it takes me hours to get like 2 or 3 sentences out. Hours. It's awful. I hate it so much. I will never do that to you. I will never make you do a Zoom, and I will never make you embed stuff into a PowerPoint. It's terrible.
You are not my favorite teacher.
Hey, that's one of the interesting things for me about having gone on this bachelor's journey this year. I just started in January, so I'm only 1 year in, is that I now have this unique perspective from a student's point of view. And I can now put myself into your shoes, and stuff that I hate and dread, I now know don't do that to you.
And so now you find yourself ironically sitting in both chairs at the same time.
I am in both chairs at the same time, and I've always been empathetic, but I'm even more so now, especially when it comes to assignments and stuff like that, because it's been an eye-opener. The nucleus, we call it the brain of the cell. It doesn't really function like our brain, but what it is the blueprint. Everything about that cell's function, what it does, how it does it, everything that that cell needs to know about everything in its world is located in the DNA. And that DNA has to be replicated when the cell replicates. And this is where we started talking the other day about like viruses that go in and mess up the DNA, or radiation goes in and messes up. Because then when that cell divides, guess what happens to the DNA? It replicates the incorrect sequence that had been messed up. So you have one tiny cell that has the DNA messed up, and now it's replicating cells with that same messed up DNA. And now you got a huge domino effect.
So it's like a copycat and duplicate. If it's a mess up,
It-
it'll duplicate. it
Every
as well.
cell will perfectly replicate the original.
Okay.
But if the original is messed up when it replicates, it will replicate the messed up. It doesn't correct it. It is a
Right.
perfect copy. Think of it like a Xerox machine.
Right, right.
So if I put a perfect copy into the Xerox machine, it comes out with perfect copies. But if I put a piece of paper in there that has a bunch of spelling errors, does the copy machine fix the spelling errors for me?
Fits it right the way it's always.
Spits it out exactly the way I put it in. That's how cells divide, and that's why it's so important to understand what sort of things can mess it up, because it's going to start copying that messed up. So it is the control center. It maintains all of the information about everything the cell needs to know. Some cells might have more than one nucleus. We talked about this, like our cardiac cells, our muscle cells, humongous cells that need multiple informational areas. And we have some cells that may not have a nucleus at all, like our red blood cells. They don't need one. They're transport only. They're kind of, I call them like a dummy cell. They don't have to think about stuff. They have one job and they do it well, and that's it. A disadvantage to having no nucleus, though, is that the way that our body produces the cells, they don't replicate by mitosis. Our bodies have to pump them out and create them somewhere else. So it's like with my liver has damaged cell and it's getting ready to divide. It divides a perfect copy and then autolysis. Red blood cells, we have to create those. We have like red bone marrow that creates red blood cells. We have a huge storage facility of red blood cells in our spleen. Kind of what our whole spleen is for. It's just simply
Like the bank.
It's a bank. Well, I call it it's the training, it's the boot camp for red blood cells. It's also the retirement center. It's like the Walmart when old people go and work at Walmart. So what the spleen is filled with is little immature red blood cells who haven't quite figured out how to do the job yet, and senior cells who are too old to do the job but not yet too old to be destroyed. And so we have this little storage facility, and that storage facility is what contracts when you need more oxygen than what you're getting in. You're exercising. Don't be a hero, though. Don't be a hero. That's why I don't exercise. It's far too dangerous. Far too dangerous. What happens to your oxygenation needs when you exercise? They go up. You need more oxygen. If you need more oxygen than you can take in, well, your little red blood cells only have a finite capacity to carry stuff, right? Once the tractor trailer is full, you can't put more stuff into the tractor trailer. So if all of my tractor trailers are filled to capacity and I still don't have enough, what can I do? Send for more tractor trailers. And so what happens is my spleen goes on it, boss, and it squishes out like a sponge and splooshes out all of these tractor trailers. Now, some of them are not quite ready to work and some of them are kind of old, but they're better than nothing. Problem is it's a one and done. When your spleen contracts, it's your body literally trying to save your life. It thinks you're about to go, and it splooshes. And that's when you get a stitch. You ever had, I call it a stitch, when you've been running
Yeah.
and you get that super sharp pain in your left side,
That's what it is.
super sharp pain in your left side, and most of your coaches are like, walk it off, walk it up. No, that's your body literally telling you, stop, sit down, you're done, because you have gone beyond what we can handle. And your spleen has contracted, that's why it's always in your left side, because that's where your spleen is.
Mind blown.
Right?
Now I know.
Come on.
Now you know. And when you start getting that stitch, you need to sit down and stop, because that's your body literally telling you, stop.
Then you get a job saying, no pain, no gain, keep it going.
Right, and that's the problem for me, because now your body is telling you, we're done, and you're asking it to push
You're
even
going on.
further, and that's when you really do some serious damage. Yeah. Well, what'll eventually happen is your brain goes, ha, you're adorable, shut everything down, boys. And it sends the message to your muscles, and suddenly your muscles are like, and you can't even stand up, you get vertigo, you fall over. Your brain's like, okay, this dummy didn't listen the first time, shut her down. Shut it down.
Find out the hard way.
Shut it down, and then you fall down, and then you are incapacitated. All right, so the nucleus, the nuclear anatomy, we have the envelope, which is the nuclear membrane that holds everything. The nucleoplasm, which just like our cytoplasm, plasm is the jelly-like structure, right? The gel. Nucleoplasm is just plasm gel in the nucleus. We have our chromatin, which is what is our DNA, and our nucleoli, which is kind of the yolk of the egg of the nucleus. Does kind of look.
And so what was the nucleus? Okay, the envelope.
So here is the nucleus. So this is the cell. This is our universe of the cell. Here at the center of the universe is the nucleus, and then this dark spot right in the middle, this yolk, that's our nucleoli.
So the chromatin is the DNA.
So think about the word chromosome.
Yeah. But isn't chroma mean like all colors?
So that's a fact. I give you a unicorn high five for that. You're absolutely right. Chrome means all colors because it reflects any other color.
But if you change up the suffix or root or the.
But not all medical terms are made of parts. In this case, chromatin is a word unto itself. It's not that we have chrome and then the suffix tin. Chromatin is its own word.
It's its own root word.
It's its own word. You can't break it down. So although, yes, you're absolutely correct what chrome means, in this case, that component is not part of this word. So our nucleoplasm, this is going to be kind of the skin of our nucleus, and it is continuous and then is an extension here into our rough ER, which we just talked about. And it's got these little holes in it, these little pores, and that's how our RNA gets out. We have the DNA inside the nucleus like the queen, and she never, ever, ever leaves the castle. But I do need to get instructions out of the nucleus. So copy it down, we all know that, the RNA, brr, 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 brr. How does the RNA get out? It gets shot out these little pores.
In other words, it goes and gets orders.
It gets its orders, and it takes its orders out of this pore, out of the nucleus, and then finds the appropriate ribosome. I'll just continue to use these words so you keep hearing them. The factory is the ribosome. Reads it, forms the protein. The protein is then encapsulated, sent to the Golgi apparatus, where it is repackaged and then sent wherever it needs to go. It all starts here in the nucleus. and the nucleoplasm is the kind of the skin of the nucleus, the shell of the egg. I like the analogy of an egg. Give you guys a minute. What is the prefix peri? There you go. So the area in between these layers is the perinuclear space, a space around the nucleus. I have got review, I know we may not finish this, I don't even know how many slides are left. Three, oh, so yeah, we'll probably finish this today, but we will spend the first 10 or 15 minutes tomorrow doing review for your test on Friday. I'll do what I did last time and give you some sample test questions and let you know the depth to which we're gonna go. Oh, don't start that.
I just get off the peppers in the like first class to my third class, but in medical technology. And so, yeah, so like being in here, not being able to drink Dr. Pepper, I'm like,
Right? I'm not allowed to drink Diet Coke, you're not allowed to
I
drink
know,
Dr. Pepper.
I feel for you.
Just saying.
Now.
Oh, that reminds me, one quick thing. I know that I announced it yesterday. You guys will be Zooming medical terminology today.
I just saw my
Yes.
email.
Oh, she's fine. Absolutely fine. So what ended up happening, I'm sure she'll tell you, she might tell you more, but she was potentially exposed. She had it was either a friend or a family member that tested positive. And so per our protocol, if you come in contact with someone that tests positive, you are quarantined until you yourself can get a test. And then once she tests negative, she'll come back to campus. So it's just our safety protocol. That's all it is. And so that's why she's Zooming off campus until our test results get back. So she's fine. She's had no symptoms.
Good.
But we have, as you guys know, very strict.
Be
It
asymptomatic.
seems like she's very strict on it too.
Yeah.
She's,
I
we all are.
yeah.
We all are. Because this is no joke, you guys. I'm not taking responsibility for one of you getting sick. Because we relax in what we were doing around here. So, yep. There's no more, unfortunately, you say, no, Miss Huff, I can't come to class today because I'm not feeling well. Okay, well, now you're quarantined until you get a test. Used to be, you could kind of take a sick day. Nope, not anymore.
They don't want free sick days.
No more free sick days. Yeah, if you are even potentially exposed to anyone,
Like any kind of sickness. Whether it be a regular cold, a child with a regular common cold.
yeah, because we can't tell the difference between a common cold and COVID. You also can't even tell the difference between like GI upset, because now they're showing up as a GI upset. So yeah, if you've got a sick anything that you've come in contact with, you need to inform me immediately, go get tested and quarantine until that test result comes back. We're not fooling around with this. Heck no. So that's all it is. But I will be setting you up. You'll just come in here and I'll set you up and she'll just be up on Zoom. So it'll be like she's here. The nucleoplasm, as I said, plasm is like gel. Think plasm gel. Nucleoplasm is just this jelly-like substance. It resembles the cytoplasm, the cytosol. And so here inside our nucleus is our nucleoplasm, the jelly-like substance. That was just a definition. So yeah, you'll be Zooming that class today and tomorrow. And then I'll let you know. Well, it doesn't affect you again until next week.
Right.
So I'm anticipating our test will be back by then. But if anything changes, I'll keep you informed.
Nice.
Can you point out the nucleoplasm again? Right here is the nucleus. Well, actually, this whole thing is the nucleus. So here's our nucleoplasm. It's just this light purple. It's just the gel that fills it up. It's like cytoplasm, but it's inside the nucleus, and that's why we call it nucleoplasm.
Yeah.
A little bit of review, DNA and RNA, composed of nucleotides. Oh Lord, there's that big word. We talked about that, it feels like a million years ago. Remember our nucleotides, can anyone tell me one of the nucleotides? Is that chromatin.
Is that adenine and guanine stuff?
Yes, adenine, guanine, thymine, uracil, and uracil. God, you guys listen to me sometimes. I love it. Which? Tell me one pair.
Adenine and
Adenine and thymine, and then
Adenine and glyamine.
right, car in the garage. Apple tree, put your car in the garage. And then in the RNA, uracil replaces what? Thymine. You too. Nice. We don't even have to do this kind of review. So when we get into chapter 4, that's when we're really going to start talking about how do we divide and replicate the DNA, kind of unzip it and then re-zip it and get 2 exact copies. But that makes sense, because if we know what nucleotides go with what, if I have my DNA, and I go zip, and I now have 2 individual strands, I know what has to bind with this one to complete it, and I know what has to bind with this one to complete it, and I end up with 2 perfect replications of this DNA. And then what happens when we start getting cell division is this super coiled DNA forms this kind of X-like structure. You've all mentally seen a picture of a chromosome. Right? It has that kind of X structure, that rounded X. Well, each half of that is a perfect DNA. And then the chromatin pulls the centrioles, rather, pull it apart tug-of-war style, and then each cell gets a perfect copy of the DNA. It's really pretty cool. Mitosis is pretty cool. I'll give you guys.
Oh, yeah.
Oh no. Well, it's a good thing my lecture's almost over. All right, I'll go back to the next slide in a minute. The nucleoli, this is where our ribosomes are created. The little factories are made in the nucleoli. They're then shot out and they adhere to the endoplasmic reticulum. That's it. That's all she wrote. Chapter 3, done and done. So tomorrow I will spend the first 10 or 15 minutes doing some review. Then we will begin chapter 4, which is transport in and out of the cell and cell replication.
Tough.
Yes?
On your website, I'm looking at the testable material PowerPoints.
Yeah.
It has a part 3 as well. Is that also part of it?
I don't know. Let me look.
Part 3 has like osmosis and all this other stuff on there we haven't learned yet.
Oh,
so.
No, that would be, so that is actually, I'll have to retitle that. That part 3 is actually chapter 4.
Gotcha. Okay.
Chapter 4 in a previous textbook used to be chapter 3. And chapter 3 was just this humongous, ginormous, amazingly large chapter.
Now they've actually.
Now they've split it into 2 separate chapters. All right, you guys.